Good morning. Today is our last day of the meeting. So we have in the program 3 activities for today. So we will begin with the award ceremony. We are going to nominate and award one student for each categories. So following, we are going to have our closing keynote, Dr. Ruslan Medzhitov. It's an honor and a pleasure to have him as our closing keynote. He made stunning contributions for the inflammatory field, innate and acquired immunity, also for inflammatory disease. So it's a pleasure to have him closing the meeting. But I will leave his introduction with the chair a little bit later. So following, we are going to make some closing remarks. I'm going to give some information about this meeting and the next meeting. So let's go to our first activity. So I invite Dr. Rosane Viana to be with me here in the screen. So I want to say that I was very impressed with our students. I couldn't watch all the presentations. The presentations were simultaneously, so I couldn't check all the presentations, but I passed by each room and I could see very nice data, very, very good presentations. The students, they discussed the results. They presented very well, and I was amazing to see the enthusiasm of the students. So I have to say, I told my colleagues, I got emotional because I think that science is still alive in Brazil, still alive in Rio de Janeiro, and we have hope. We have hope, and it was a great surprising. So I will pass the floor to Rosane. I think it's more than fair for her to present or to announce the winners. She was 100% involved in the selection, organization, evaluation. So I think she deserves to present, to do the announcement. So Rosane, the floor is yours.
Thank you, Claudia. First of all, thank you for inviting me for this award ceremony. It was a real joy, as it was to participate in the workshop. You got us all involved, and it was a special joy to deal with the works from students, because we had many submissions, like I believe 85. Very, very nice works. So we had our team working to first select and nominate honorable mentions, and we had 24 honorable mentions, 6 in each category. And sometimes it was very hard to pick these 6. So I really congratulate everybody who sent their abstracts. Really, the workshop was made by everybody who could engage and participate, and it was a great engagement, and we were very happy about it. We are struggling with many difficulties in science, but it's really rewarding for us to see that the enthusiasm is still on. So, and it was a special joy for me to really be through the whole stages and steps of that and deal directly with the students. So as Claudia was saying, we had 4 categories: the undergraduate students, the master students, the PhD or doctoral students, and the postdoc fellows, and 6 nominees in each category. So I first of all, we invited them, some of them to actually we invited all of them. They will appear here so that they can, the real actors of this moment, can celebrate with us. So maybe if they show up a little, if you can see them. So the undergraduate students who were honorable mentions and were able to present their oral communications were Fernanda Neves da Cunha, 𝗬𝗲𝗂𝗶𝘁 da Silva Albertoni, Matheus Macedo Monteiro, Nicolas Mateus Pereira de Souza, I apologize, there was a mistake there, and 𝗕𝗲_Henrique de Lima Vasconcelos. So congratulations to you all. And the award goes to Cláudia.
Congratulations, Fernanda.
Congratulations, Fernanda. Congratulations to you all, guys. You did a great job. Thank you for participating.
Thank you.
So now I ask the undergraduate students to move to leave the room. Unfortunately, I think I can ask Fernanda to stay until the end, and we'll have the master students. So the master students who were honorable mentions were 𝗕𝗲𝗂𝗃𝗃𝗀𝗧, 𝗕𝗲𝗂𝗃𝗃𝗃𝗀𝗃, 𝗕𝗲𝗵𝗂𝗃𝗃𝗃𝗃, 𝗬𝗵𝗂𝗃𝗃𝗃𝗃𝗃, I hope I am pronouncing it right, 𝗬𝗲𝗵𝗂𝗃𝗃𝗃𝗃 Francescantoni, and Vinícius Amorim Beltrame. Are they all there? And the award goes to Cláudia.
The Oscar goes to...
The winner is...
The winner is... we should think about the music here.
Yes, unfortunately we...
Congratulations. So she's not here. But...
I'm afraid Esther could not come today, but and I don't have a photo of her, so I apologize for that, but we can all congratulate her later. If she's watching us, and she wrote that she was on an experiment this morning, so I hope she has some minutes to show up.
Okay,
so
thank you very much.
Congratulate you all, girls. Thank you for participating. And now we'll have the doctor or PhD students. Again, we had 5 oral communications.
If they are here.
I'm afraid Gustav's saying that nobody came into the room. Let's wait a few minutes. Maybe they're joining, but I hope they're watching on YouTube. I let them warn that there was this move. So, okay, so some are coming. So our honorable mentions are Beatriz Bastos de Moraes Tomasi, Felipe Saciano Lezer, Grace Nascimento Pires, Isadora Ramos de Andrade, Jesuíno Rafael Machado Ferreira. Congratulations to you all. And the award goes to Cláudia.
Tan, 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 tan.
Isadora de Andrade.
Congratulations, very nice.
Congratulations, Isadora.
Thank you.
So everybody's working today, you see?
Thank you very much.
Thank you all for participating, and congratulations for the great work, okay? And now we have the postdoc. I don't know if there's anyone in the room. We can wait a few moments until they come.
I didn't say anything.
Okay, I can wait.
You can.
I can name them. So we had 5 honorable mentions. Actually, there were 6 honorable mentions, but 5 oral communications. Luana da Silva Chagas, Luciana Tavares, 𝗬𝗲𝗂𝗃𝗃𝗃𝗃𝗃, I apologize again, Marina Valente Barroso, 𝗕𝗃𝗂𝗃𝗃𝗃𝗃. So Luana is there, Luciana. Anybody else? Is anyone coming? Okay, Cláudia, so we can announce the award goes to Luciana Tavares.
Congratulations.
Congratulations. So maybe
Thank you so much. It's such a pleasure and an honor to be awarded in such a good meeting. Thank you all so much.
Thank you. It was great.
Thank you, dear. So let's see the winners, if they are still there. I don't know if they left the room. Fernanda. Isadora, if she's still there. Luciana, and I don't know if Esther could come. So we had 4 women winners. Congratulations, girls. But I also want to acknowledge, so thank you, girls. Well, you can stay, but Claudia, I will move to the next slide, please, so that I can thank and acknowledge the referee team who worked with us. We had 2 professors in each room, but in addition to these 8 names, we also had the help all the time from the scientific committee of the workshop, who worked doing the first selection of abstracts. So I want to thank and acknowledge Cristiana Bandeira de Mello, 𝗬𝗹𝗂𝗃𝗍𝗔 de Moraes, Leonardo Travassos, Manuela Lanzetti, Mariana Renovato Martins, Priscila Olsen, Rodrigo Tinoco, and myself, who had the joy and the blast of working with these students. And last but not least, I also want to acknowledge and congratulate the 3 top abstracts. These are the 3 abstracts that are getting most views. Is that right, Claudia?
Yes.
So we could not have a poster session, but the abstracts are available at the website, and you can access them and read and get in contact with the authors. So we really exhort you to do that. And these 3 authors are getting the higher views. So thank you. It's obvious that you had great work. Congratulations, Gilson Dornelles, Isadora de Andrade, and Marina Valente Barroso. And thank you all for participating.
So I would like to congratulate again the students. Probably they made part of the data during pandemic with shortage of funding and in a, how can I say, in a strange scenario that we live today. So it's, of course, it's top. They did nice and very good job. So all the students that we selected. So thank you to participate, to be with us, and I hope to see you in the next meeting. Rosane, we didn't tell.
I'm sorry. I think
Right?
I forgot to tell that all the awarded, they gained as a prize 500 reais, and also the pre-registration for the next workshop, which will be next year.
Next year
in Rio.
Presential in 2022. So I hope to see all you there for the 4th workshop on inflammation. So we still have some minutes before the next session. So we will be off, but please stay connected. We'll be back right soon. Okay, so keep an eye on the screen. We'll be back.
Stay tuned.
Stay tuned.
Good morning to you all, and welcome to the closing keynote session of the third workshop on inflammation. It's an honor to be here and to continue the celebrations of the 70th anniversary of the Biophysics Institute of the Federal University of Rio de Janeiro. 75 years ago, the creation of the Biophysics Institute helped establish scientific research as an integral part of the Brazilian university. Today, universities in Brazil are responsible for about 90% of the national scientific output. I would like to thank the third workshop on inflammation and its organizing committee for the opportunity to join to host Dr. Ruslan Medzhitov's conference on the evolving perspectives on inflammation. To introduce Professor Ruslan and chair this session, we are going to have Professor Marcelo Bozza from the Institute of Microbiology at the Federal University of Rio de Janeiro. Marcelo, you have the floor.
Thank you, Bruno. So it's a privilege to have Ruslan Medzhitov in this closing lecture. So Ruslan is professor of immunology of Yale, and 𝗕𝗃𝗀𝗃𝗃, a scientist. So everyone that works in the immunology field knows Ruslan, but I would like to point out some important discoveries that Ruslan made over the past 30 years, more than 25 years. So Ruslan discovered TLRs, and in this subject, he demonstrated the role of TLRs on DC maturation and its importance on adaptive immunity activation, working as adjuvants. The role in differential cargo of antigen proteins, and the role of subcellular compartments on TLR signaling, and a beautiful work on the role of TLRs on mucosal homeostasis. And then he went on and it has been doing very interesting work on allergy, demonstrating the role of basophils triggering the naive T cell response, so priming T cell response to a Th2 phenotype, and also proposing interesting ideas such as allergy as a defense mechanism. So we have this idea that allergy is only pathological, and Ruslan has been proposing that is part of a defense mechanism. And in the same line, he proposed that food allergy is a type of quality control for obtaining the food. And then in macrophages, he has made very important contributions in terms of transcriptional programs, the ontogeny of tissue macrophages, the role of tumor environment in defining the macrophages, and vice versa. And the idea that macrophages play a major role in tissue homeostasis. And then, you know, he also has been pushing the idea about the host defense mechanisms, not just related to resistance, but also the counterpart, that is disease tolerance. He has done both experimental work as well as theoretical propositions that are very important to the field. And then we come to the main subject of today, that there is inflammation. And again, the idea that inflammation, it's an essential part of the tissue homeostasis. And all these different subjects, they have been immersed in evolutionary perspective. So I think that very few scientists have a similar contribution, and it's really privilege to follow Ruslan's work. And finally, I think that he has another associated great influence as a mentor. So I think that these two things I consider the most important things besides his generosity and friendship. So Ruslan, it's really a pleasure to have you here. And I hope to see you soon in person.
Obrigado, Marcelo. Thank you so much. Thank you for inviting me. Thank you for a very kind introduction. I really hope to visit in person soon, because Brazil is my favorite place to go. Marcelo knows that, so I'm not exaggerating. Okay, so I will, Marcelo, if when I start sharing, if you can just say okay or not. Yeah.
Yeah.
Good.
You're
okay.
We are all
right. Thank you. So yeah, it's a great privilege to talk to you about our work and our perspectives on inflammation. I gave this strange title because that's where I am mentally. I think our general perspective on what is inflammation is currently maybe at the stage where it needs some major revisions that would make these views more general. And I'll try to explain why. So I will spend the first 10-15 minutes talking about that, because I feel that this is, to me, this is the more important point than showing some data. But then in the end, I will use a couple of stories that kind of illustrate one aspect of this framework that I'm thinking about. So as you all know, the history of modern understanding of inflammation goes primarily to these two scientists, Rudolf Virchow and Eli Medzhitov, who were contemporaries. And as you know, the view that inflammation is this very fundamental part of defense system was pioneered by Medzhitov at the time when the views of pathologists dominated, especially proposed by Rudolf Virchow, that inflammation is primarily a pathological process. And Virchow had much more authority at the time, rightfully so. He's one of the greatest scientists of the past few centuries. And that view predominated. And until now, inflammation is taught and thought of primarily from the perspective of pathology. So even at medical schools, inflammation is taught as one chapter in pathology textbook, and a little bit in immune system. Of course, we appreciate inflammation. But I want to emphasize that Medzhitov thought of inflammation as a sort of being on a spectrum from what he called harmony-disharmony balance, which is in modern terms what we call in homeostasis. At that time, the term homeostasis wasn't yet coined until 1929, when Walter Cannon proposed that term. But it's the same thing. Harmony-disharmony balance is exactly what we call homeostasis now. Then he suggested there is physiological inflammation that participates in various body functions, and then there's pathological inflammation, and finally infection-induced inflammation or immunity. And this was actually very important insight that was pretty much ignored until recently. But the importance of this is to highlight that what we normally call inflammation is something that is here at the extreme end of the spectrum. And I think the genius of Medzhitov was to realize that there is a whole spectrum of biology that goes all the way to what we consider normal physiology, homeostasis. And that view, I think I'm very much subscribed to that view that he proposed. So the problem is that if we think about what causes inflammation, the textbook answer is that every textbook will say inflammation is a response to infection or injury. yes, inflammation is induced by infection or injury, for sure, but there are many cases where inflammation is present in the absence of infection or injury. We all know that. Examples would be obesity, aging-associated inflammation, or cancer-associated inflammation. There are many conditions where there is inflammation, but there is no tissue injury or infection. And so the question then is, what causes inflammation? Infection and injury is just one answer, but it's not full answer. And if we think of all the different situations that are associated with inflammation, the common denominator for all of them is that there is some kind of a problem in the system. Of course, injury is a problem, infection is a problem, but how do we more generally describe what kind of problems cause inflammation? So I've been thinking about this the last few months, and I suggest this view. This is the first time I'm presenting it, so you can please criticize and give me your feedback. But my view is this, that the question is, what kind of problems a system can experience? If we agree that inflammation is induced if there is some problem, then what kind of problems can affect the system? And I think that there are three types of problems that can occur in any system, both evolved biological system or engineered systems. The three types of problems is the loss of structure, or what we would call damage, tissue damage, loss of function, or loss of regulation. And structure, function, and regulation is a key general characteristics for any system, right? And so I think that all three types of problems, loss of structure, function, and regulation, can cause inflammation, but cause different types of inflammation. So at the first approximation, again, this is a very much kind of evolving view. It's not, I'm sure there are many things that would need to be adjusted as we go forward. But at first approximation, I consider the following type of classification. That loss of structure is what causes type 1 inflammation. It's the type of inflammation that is classical canonical inflammation that leads to vascular response and cardinal signs of inflammation and so forth. So this is what's caused by microbial infection and tissue injury. And the type 2 inflammation, I think, is caused by loss of function. And type 2 inflammation is what's also called allergic inflammation. And the reason I think it's caused by loss of function is because if we think about what are the common features of allergens and the types of adjuvant effect that is associated with allergens, I think that the common feature here is that they are not inducing damage necessarily, but what they do, they interfere with various biological functions. And they can do it in a variety of ways. So this is a very much unexplored area to understand how loss of function leads to allergic inflammation. And importantly, we don't know which functions are particularly important here. Presumably not all functions are designed to be coupled with inflammation, but many are, especially many barrier functions. And I'll show you an example. And then the third type is a loss of regulation, and that what I would refer to as physiological inflammation. So this term that Medzhitov originally proposed has been used kind of to refer to inflammation that occurs in the absence of infection and injury, and inflammation that doesn't have cardinal signs of inflammation and so forth, but it's important in a variety of contexts, like, for example, in thermogenesis. There is an involvement of ILC2 cells, eosinophils, IL-33, IL-4 in inducing beiging of the fat and important for thermogenesis. And there are many other conditions like loss of glucose homeostasis and so forth. Many situations where loss of regulation would be associated with an inflammatory response. So just schematically, we can think of loss of structure is the easy one because that's the most familiar. That's tissue damage. When there is damage, we know that there is either release intracellular components like the ATP, histones, HMGB1, there's various chromatin-associated proteins, nucleic acids, all these things that together somehow trigger inflammatory response. We don't understand all the molecular details, but it's very clear that tissue damage causes inflammation. It can also be due to desequestration if there is loss of some barrier, either endothelial barrier or intestinal epithelial barrier or skin barrier and so forth. There's desequestration that also results in inflammatory response. So that's the obvious one. The next one is the one that I suggest as a new type of cause of inflammation is a loss of function. And this primarily came from our sort of studies and thinking about allergic inflammation. And what happens often that in the case of food allergies, for example, is that the normal digestive and absorptive function in the small intestine, where we have brush border enzymes, pancreatic enzymes, and some, let's say, nutrients, some protein get digested and absorbed. What often happens in the context of allergens is that there is some kind of interference with these functions. For example, many plant secondary metabolites that are noxious to the organism, they're noxious because they interfere with digestive function. And then you have accumulation of undigested protein, translocation. So that would be just one example. Another example could be in the airways when we inhale large particles, like large pollen particles, dust particles. Normally, the particulate matter is handled by ciliary elevator in the airways. But if they are too big to handle by ciliary elevator, it interferes with normal airway function, and that also can trigger reflexes like coughing or sneezing. But also we have preliminary indication that it also results in allergic inflammation. And finally, loss of regulation. In this context, it's primarily means loss of homeostasis. There are different types of regulation, but it's a loss of homeostasis that's the most obvious case here. So schematically, I showed here in the green color indicated normal homeostatic control. So just the system deviates from set point, and homeostasis operates based on negative feedback and brings it back. If you go up, it brings it down, and so forth. If this could be glucose, this could be body temperature, blood pressure, or whatever it is, homeostasis keeps it in the right range. But if the perturbation to the system is too strong for homeostasis to handle, and if the system goes outside of homeostatic range, that's where I suggest that inflammation is induced, and this would be physiological inflammation in this classification, and the goal of that inflammation to bring it back into homeostatic state. So these are three types of conditions I suggest that associate with three types of inflammation. And the second point I want to make is that whenever we have these three types of perturbation, the system attempts to both get rid of the cause of inflammation, but also to adapt to it. And the adaptation happens by forming structure function regulation that are complementary to the basal structure. So let me explain. So this is the basal structure of an intestine. So you have normal sort of composition, epithelia, you have some eosinophils, some mucus, and so forth. So it's a normal state. But in response to inflammatory stimulus, infection, or it could be allergy, there's an alternative structure. There is a remodeling of the tissue. Now there is a lot more goblet cells, for example. There is a much more mucus and more eosinophils, and there are many other things that change. So it's alternative structure, and this can be called inflammatory structure, and this is basal structure. Similarly, we can consider inflammatory function. So if this is a normal function of small intestine, normal peristalsis in response to food matter in the lumen of the intestine, activates enteric neurons, promotes peristalsis, and so forth. But on the inflammatory conditions, we have a different mode of function. We can call it inflammatory function, inflammatory peristalsis, where now histamine released by mast cells or serotonin released by enterochromaffin cells induces massive contraction of smooth muscles and peristalsis, leading at the extreme cases to vomiting or diarrhea. But the point of it is not to optimize digestion absorption like here, but to get rid of whatever caused the inflammation, to expel it from the body. And finally, an example of inflammatory regulation would be when we have normal control, let's say, of lipolysis, just randomly picked one function, the one type of regulation. So regulation of lipolysis happens primarily by sympathetic neurons, as well as some other hormones. And so this signal, like norepinephrine, would induce lipolysis, release fatty acids in response to fasting or cold. But under inflammatory conditions, now inflammatory signals take over, and they can induce lipolysis regardless of the state of sympathetic nervous system or cold or fasting. It doesn't matter. It will override that. And so that's what I would refer to as inflammatory regulation, where inflammatory signals, instead of homeostatic signals, perform the regulation. A different way to look at inflammation is from this perspective that I used for a few years now, were considered by causes of inflammation and their purpose and pathological outcomes. So infection and injury, again, this is type 1 inflammation. The goal here is defense from infection and tissue repair. And there are corresponding types of pathological outcomes of if inflammation is excessive, like sepsis, autoimmune disease, collateral tissue damage, fibrosis, and so forth. When it's noxious substances, non-infectious noxious substances like toxins and so forth, then the goal of inflammatory response is expulsion from the body and increased barrier function. And that would be allergic inflammation. And the extreme, it leads to allergy. And as Marcelo mentioned, that I think that allergic inflammation is basically, it's a part of a defense from noxious substances. It's just when it's extreme, exaggerated, then we call it pathological allergy. And finally, there is another type of a cause of inflammation that is just modern lifestyle. It's unnatural aspects of our lifestyle, too much, not enough movement, too much calories, salt, alcohol, whatever, sitting, watching TV, these sort of things. And so there is no physiological purpose for inflammation induced by this, because this is what can be thought of as loss of normal homeostatic control. And the consequence is primarily what is driving most diseases of modern civilizations, which is metabolic, cardiovascular, and aging-associated diseases. So I would consider this is inflammation caused by loss of regulation, this is loss by function, and this is loss of structure. So all types of inflammation can be viewed as this type of inflammatory pathway that has 4 universal components: something that induces inflammation, like pathogen or loss of homeostasis, tissue damage. Then there are some specialized sensors like macrophages, mast cells, and so forth. They make various inflammatory signals, and they affect different target tissues. So that's the sort of general view of what inflammatory pathway is. There's always these 4 components. And notice that this view is exactly parallel, it's a version of a view of any type of control system, where you have, for example, something you want to control, like body temperature or room temperature or whatever. There is always you need sensor to know what that temperature is currently, or whatever that value is. And in the case of room temperature, it would be thermostat. And then you need something that can change that temperature in the right direction. That would be the effector, like furnace or air conditioner. And then you need signal that connects them. And then the point of the effector is to change this in the right direction, whatever the variable is. Similarly, systemic homeostasis, we have, let's say, blood glucose homeostasis. We have sensors, alpha and beta cells. We have effectors, liver and muscle and fat, and we have signals, insulin and glucagon, and they act to control this value. And similarly, with noxious perturbations like pathogens, toxins, and so forth, there are also sensors like macrophages and so forth. There are now inflammatory signals instead of homeostatic signals right here. And the effectors would be granulocytes like neutrophils, eosinophils, and as well as other types of cells that their point is, again, just like for effectors here, is to deal with whatever caused the response. So if it's a pathogen, then it's eliminate the pathogen. If it's tissue damage, then repair the damage. Okay, so this type of design is common to all these systems. So this is the sort of a very generic control circuit. And what's interesting, if we look from this perspective, is if we compare homeostatic circuit and inflammatory circuit, is that the effect of cytokines is not only to activate effectors or to recruit effectors, but cytokines also act on homeostatic sensors and effectors. For example, TNF will act on beta cells and suppress insulin secretion, and TNF will act on liver and muscle to cause insulin resistance, to make them less responsive to insulin. So cytokines, inflammatory signals, can override the effects of homeostatic signals, and that's a common theme in inflammation. And the reason for why cytokines are dominant is because they have higher priority. If this is controls when things are normal, these are induced when things are not normal. And of course, if they're not normal, you have to take care of it. And then you often do it at the expense of the normal homeostasis. And finally, in addition to what I want to highlight here is that in addition to the effectors of inflammatory response, inflammatory signals act on many tissues and organs that are not effectors. They don't participate directly in dealing with whatever caused the either environmental condition or loss of homeostatic control. And these non-effectors participate in a very different way. Whereas if maybe I have a better picture here. So let's say if there is an infection, sensors like macrophages will make inflammatory cytokines, and they will act on effectors like neutrophils, and they will provide defense from infection. And this would operate as a negative feedback loop, because neutrophil get rid of bacteria and now you stop the response, right? So this is what's called the resistance arm of the defense. So it's eliminating the pathogen. But in addition, we know that cytokines act on all organs, on the brain stem, on adipocytes, on liver, on kidney, intestine. Everything is affected by cytokines. But these organs don't directly participate in eliminating pathogen. And then the big question becomes, what are the reasons for cytokines to act on all these organs? There may be more than one reason. And what is the logic of this regulation here? Here we know, because you just need to activate these cells to get rid of infection. But what is the logic here? And that's a big question. But I want to just emphasize here that the way that this arm of the response works is very different from this. So here it's a negative feedback, and here it's a feed-forward loop. And that's what promotes adaptation to these conditions of infection or whatever caused inflammation. So this resistance versus adaptation is the different way. There are different terminology to refer to the same concept. In infection biology, it's called resistance and tolerance, but it's the same idea. So just to remind you, the resistance and tolerance are two types of defense strategies, and it refers to the way that system can react, in this case, to microbial infection. So low microbial infection, high health. This microbial infection, health goes down, and then you can either come back by resistance, by getting rid of microbes and you become healthy again, or you can do it by increasing tolerance, that now you reduce the cost of infection and now you're still healthy even though there are microbes. And that would be example with, for example, commensal microbes, or examples with some species like bats that harbor many viruses that are very dangerous to primates, but harmless to bats, right? Like coronavirus and Ebola and dengue and so forth. So there is this phenomenon that is still poorly understood in terms of mechanisms, and needs to probably will take long time and many people to fully understand it. But in addition to microbial challenge, this part could be also inflammatory response. So when there is inflammatory response, health goes down. So you go down here. then you can either regulate inflammation, downregulate it, and go back to healthy state, or there could be increased adaptation to inflammation. And then you go here while there is still a strong inflammatory response. And in the end, I will suggest one concept how this might work. There is no specific data yet, but it's just an idea how this might work. Okay, and a couple final points is that all inflammation, so the reason I put inflammation at the expense of health here is because we all know inflammation always comes with a cost. And the question is, what different types of costs that inflammation causes? And I think there are two types generally. And this is a little bit heavy wording, but it's a simple idea. So first, this one. This is on the right side is that's what we're all familiar with. There is always collateral damage. So collateral damage is not something we need or we want, but it's not intentional, but it's unavoidable. If neutrophils are activated, they will cause some mess, right? So that's collateral damage, unavoidable and unintentional. The other one is more interesting. This is an intentional suppression of some functions that are incompatible with the goal of inflammation. So these other functions, if they have lower priority than inflammatory response, and they interfere with, they're kind of standing in the way of inflammatory response, they will be suppressed. And that is also a cost of inflammation. And this is intentional. So this follows specific logic. This doesn't follow logic. The collateral damage is just something we can't avoid. This follows specific logic, and we really need to understand what that logic is, because this is done by design on purpose. But both of them then contribute to inflammatory diseases because both of them are causes of inflammation. And if we think about that, the cause of inflammation, we can think from this evolutionary perspective of cost-benefit of the trade-offs between the benefit of inflammation and the cost of inflammation. And for any biological trait, we can consider this type of relation between benefit of the trait and the cost. In the red triangle here, the cost is more than benefit, and such traits would be eliminated by natural selection. So this is unstable. This will be gone. But when benefit is higher than the cost, then it will be selected. From evolutionary perspective, it's fine. It's good. You benefit more than you pay for it. And all of these traits will be selected, including the traits that are over here, which provide very high benefit but come with very high cost, right? So from evolutionary perspective, this is over here is totally acceptable in the right corner. I hope you can see my cursor. But so over here, for evolutionary perspective, is fine. But if we look at from patient perspective or doctor perspective, this is what we would call disease. So anything that beyond some level of the cost, we call a disease. You infected, you have fever, you feel sick, headache, and so on. We call it a disease. But this is just the cost of inflammation, right? Evolution kept this type of system because it provides very high benefit of protecting from infection. So from patient perspective and doctor perspective, we call this health and we call this disease, but this is not true disease over here. This is the cost of the response. And it's evolutionarily acceptable. So some diseases are because evolution, from evolution perspective, it was fine. You suffer, but you survive. But because of that, there must be mechanisms that evolved also to keep us alive when we are over here. And this would be what doctors would call critical conditions. And I don't know how many of you are doctors and who treat with critical patients, but there is very little understood about biology of critical illness. Most of the critical care is sort of supportive care. And I think that this is something that we all need to change and try to understand the biology behind the survival biology that kicks in during critical illness. And that's what we became interested in that. And a good example of that, and under critical illness conditions, often you will have severe inflammation. And so inflammation is a good way to start to look into that. And one type of a very familiar example of this survival behaviors that is not necessarily extreme case, but very common, is what's called sickness behaviors. We all experience that. If you have any viral infection, acute infection, you always have this loss of appetite, social withdrawal, fatigue, sleep. You sleep 16 hours a day instead of 7 hours, and so forth. Cessation of grooming, suppression of libido, and so forth. Some of them are better, and this kind of we can explain. Suppression of libido is easy because inflammatory signals suppress gonadotropic axis. Sleeping patterns, less clear, but there is certainly alteration of circadian rhythms and probably adenosine system. Loss of appetite, not understood in details how that happens, interestingly, but in general, there is activation of some brake circuits in the brainstem that suppress appetite. Cessation of grooming, I would say, is the most enigmatic, because this is not just humans like this lady here, but even flies do that. All animals studied, when they're very sick, they stop grooming. And grooming is important for all animals because it's probably adaptive to all animals. So that's a strange one, why there is suppression of grooming. But anyway, so I'll just show you a few examples of how we approach this. Some of this is published, but it doesn't matter. It's just for illustration purpose. We started with trying to understand suppression of appetite or anorexia. And one thing we found is that infection-induced anorexia it's a very different state compared to starvation. So in both cases, the animal or human doesn't eat, but the metabolic state is very different. So we wanted to study what the role of anorexia is, and we repeated some experiments that were actually done in 70s initially, that we infected mice with listeria, they stop eating, or inject LPS, they stop eating. And then if we force feed them in the context of infection, then they all die. And this is LD50 dose of infection. This is control, and this is we give them food that they give to critically ill patients, at least in our hospital. This particular food. If we give them LPS and then 𝗕𝗏𝗃, again they die with the food. And the reason for this death was because of glucose. So we find that if we just give glucose, they all die. If we give 2-deoxyglucose to suppress glucose metabolism, they all survive. And this was not because of the effect on cytokines. They're all the same. This was because of inability to turn on fasting metabolism, because the survival was entirely dependent on a master regulator of fasting metabolism, is a nuclear receptor called PPAR-alpha, which is expressed in the liver and kidney and is responsible for fasting adaptation, in part by controlling ketogenesis in the liver. So beta-hydroxybutyrate, one of the main ketones that is made under fasting conditions, is completely gone in these mice that are PPAR-alpha knockouts. And actually, this is why they die here in the absence of PPAR-alpha, because we found that ketones were critical for survival. And what we found in this was an interesting example for, I don't know, for kind of a teachable moment for me, that these experiments were done by very talented couple of people in the lab, and one of them is a clinician who actually sees ICU patients. And what normal, like I would never notice, maybe most postdoc would never notice, but he had some extra sense sort of to look for things that are other people wouldn't. And he noticed that mice, before they died, they always have convulsion when they died from sepsis. And we thought, okay, fasting metabolism and ketogenic program both required for survival. And when mice die, they have convulsion. But ketones are also used, ketogenic diet is used to treat epilepsy. And we thought, well, maybe this is related. Can anti-epileptic drug protect from sepsis? So we used two drugs, Keppra and valproic acid. And interestingly, valproic acid could very potently protect from death from sepsis, whereas Keppra didn't. Both of them are anti-epileptic. And the reason it's interesting is because the mechanisms of actions are very different, and valproic acid is thought to work, in part at least, by inhibiting ＨDAC enzymes. And the reason that's interesting is because beta-hydroxybutyrate also inhibits HDAC enzymes. So we think that what happens here is that HDAC's inhibition, probably important for GABAergic inhibitory interneurons, which are the ones that are malfunctioning, causing the seizures, and valproic acid can protect from it. And this is a proved drug that can be used for, tested for these purposes in clinical trials. And so this valproic acid, importantly, was protective even in the absence of ketogenesis. So even in PPAR alpha knockouts, it could rescue PPAR alpha knockout, suggesting that this is our sort of way we look at it, that there is a metabolic circuit. So it's just one of probably many, where induction of anorexia causes you stop eating, glucose level goes down, insulin goes down, and that releases the brakes on lipolysis and ketogenesis. And then beta-hydroxybutyrate was important for protecting neurons from damage, and that's basically these inhibitory neurons in midbrain were the weakest link of the body. That's why mice die. Of course, animals or humans can die for other reasons, and then the weakest link would be something else, perhaps. And that's why I'm emphasizing that this is one of the circuits that provides this kind of protection. Of course, if glucose level is way too low, it also would be bad. And again, actually, Miguel Soares at the Gulbenkian Institute had a very different study where looking at the opposite end of the spectrum, where there is no ability to make glucose by liver. And then you have the opposite problem of too little glucose, which also can be lethal in sepsis or in any conditions. So this is an example where we see that inflammatory signals induced by infection both activate effectors, but also act on tissues like liver, adipose, and brain and kidney to promote adaptation to high inflammatory state. And that adaptation actually critical for survival. As I showed you, there is no effect on the inflammatory response itself or on pathogens, but there is a strong effect on survival if these tissues respond appropriately to inflammatory signals. So their goal is not to get rid of infection, but to promote tolerance and survival. And we asked, in addition to inflammatory signals like TNF, are there some dedicated signals that are specifically specialized on promoting physiological adaptations? And the type of signal we focused on initially is GDF15, which is the very interesting hormone that is induced by all kinds of conditions. Most of them are noxious conditions like chemotherapy, cell stress, infection, but also it's produced by placenta in pregnancy and in humans, not in mice, but in humans, and it causes morning sickness, for example. It causes nausea, anorexia, and so forth. Very interesting thing. And it's induced, we were looking what is induced in macrophages to look for this type of a signal, and GDF15 was one of the strong signals, and so we decided to look. And at the same time, there was also 4 papers came up to find the receptor for GDF15. All came from pharmaceutical companies because they're interested to use GDF15 as an appetite suppressor. Because there are too many overweight people in the world, and this is what they're interested in. And we thought, oh, maybe GDF15 it participates in infection-induced anorexia, and turned out that it's not. So even without GDF15, if you infect mice or give LPS, they stop eating. So it's not necessary for that. But at the same time, the only known receptor for GDF15 is expressed in area postrema, which is part of the brain stem that is responsive to noxious signals in the bloodstream, toxins, but also cytokines and hormones like this. And so what we did, we asked whether GDF15 is still participating in this adaptation because it's induced by infection and there's a high level in circulation. And so we used a neutralizing antibody and found that if we block GDF15 in the context of inflammation, then the mice all die. And this is not because it has an effect on inflammatory signal, TNF, interferon, all these IL-6, they're all normal. but still they die without GDF15. So that suggested, well, it maybe functions not as an immunoregulatory molecule, but maybe as something promoting tolerance to inflammation. If we use infection model, either sickle, ligation puncture, or flu, again, blocking GDF15 results in increased mortality. But it doesn't affect pathogen load, again, suggesting that it participates in some kind of tolerance induction. And conversely, if we give GDF15, that it promotes survival of these different conditions, infection or LPS. And that suggested another physiological circuit for survival. And in this case, it's mediated. I didn't go through all the details because they were published, but the details I feel like not super important. What's important is that there is the idea that there are these dedicated circuits that are promoting physiological adaptation to extreme conditions. This is what we can call biology of survival of extreme illness, right? This is just an example. I think this is tip of the iceberg. This is one example where hormone like GDF15 acts on 𝗂𝗵𝗂𝗶𝗃, and what we found, it actually activates 𝗂𝗵𝗂𝗶𝗃. It's necessary. So when you give LPS to animals, 𝗂𝗵𝗂𝗶𝗃 lights up, so it gets active. If we block GDF15, it knocks it down, it goes away. So it shows that this, at least the effect mediated by 𝗂𝗵𝗂𝗶𝗃 are completely dependent on GDF15. And downstream circuits also are partially dependent. Activation of parabranchial nucleus and lateral hypothalamus and amygdala, they're all partially dependent. But 𝗂𝗵𝗂𝗶𝗃 are completely dependent on GDF15. But then one outcome of activation of parabranchial nucleus is activation of sympathetic output to the liver. And that results in norepinephrine-induced promotion of triglyceride production. And we found by doing lipidomic, metabolomic analysis of the serum is that triglycerides specifically were reduced in GDF15 blockade during sepsis. And we also found that there was a strong cardiac damage. And when we replenished triglycerides, then cardiac damage was protected. And if we block adrenergic signaling using beta-adrenergic receptor knockouts, then the protective effect of GDF15 was gone. So all this, I'm not showing those data, but it all to demonstrate that there is this type of circuit in play where sympathetic stimulation of triglyceride production protects heart from inflammatory damage. And this is, again, an example of adaptive physiology to critical illness. And again, I emphasize that this is just one more example, but we don't know many examples yet. So, but there are many more that would need to be identified, because this is, I think, just a tip of the iceberg. So this suggests that there are two types of signals induced by infection. Some signals that are we more familiar with, inflammatory cytokines and so forth, antimicrobials, and they participate in immune resistance. And other types of signals, one example of that, which would be GDF15, promotes physiological adaptation and tolerance. And again, there are probably many other signals that are in this category. They don't participate directly in resistance, but they promote survival. And these types of signals would be super important interesting to identify, and probably important clinically, because they can be administered without interfering with immune resistance. If you block TNF, you have a cost of now you cannot deal with infection, but these types of signals presumably can operate without interfering with resistance. So this, again, suggests that there are two arms of the response. One is this defense, resistance mediated by cytokines, and other tolerance. GDF15 is one example. These signals act on effectors and non-effectors, and these signals primarily act on non-effectors, the ones that participate in the adaptation. And my final point is that I mentioned in the beginning about how do you reduce the cost of response. And this is, at this point, speculative slide, because we only have some initial evidence for this, but what I want to share with you anyway. So as you know, almost all the research on immunoregulation, control of inflammation, all of it has to do with studying so-called anti-inflammatory signals. Anti-inflammatory signals can be defined as signals that control magnitude of the response, how much inflammatory signals produced. Examples would be IL-10, 𝗕𝗂𝗂𝗃𝗃, TGF-beta. They all suppress production of inflammatory signals. But this type of control is insufficient by itself, because if you have an infection and you need, let's say, 10 units of TNF, but 10 units of TNF maybe will be lethal, then what do? You cannot control, people say, oh, there is a right balance between, the positive and negative effect, but there is no right balance. If you need 10 units, but it's lethal. If you make less than 10 units, you die from infection. If you make 10 units, you die from inflammation. There is no balance in this situation. And this situation is not hypothetical. It's probably very common. So therefore, I thought that we must have another type of control strategy. And that's what I suggest as a counter-inflammatory control. So counter-inflammatory signals, unlike anti-inflammatory signals, they don't control magnitude of response. So magnitude of response will stay the same. But what they do is they act on the target tissues instead of the sensors of inflammation. So in response to inflammatory signals, target organs transition from one state, which is normal homeostatic state, to inflamed state. So for example, for liver, completely changes protein expression, secretion, metabolism, and so on. The same for kidney, lung, any organ. In response to TNF, IL-1, IL-6, they transition to alternative, what we can call inflamed organ. And being in an inflamed state is what basically results in the cost of inflammation. If you're in this state for long, that's what results in symptoms of illness, and the extreme can result in death. And what I suggest is that there exists another class of signals that are called counter-inflammatory signals. What they do is that they reverse this transition to the normal state. This type of effect, when one signal acts in one direction, another signal in the opposite direction, is very common in physiology. As you know, this is what Walter Cannon proposed almost 90 years ago, 80 years ago, as what he called antagonistic control, which is common in regulation of homeostasis, like sympathetic, parasympathetic nervous system, insulin, glucagon. These are all examples of antagonistic control. And there is no reason to think that this is somehow excluded from inflammation. It actually would make a lot of sense to have antagonistic control in inflammation, where the transitions to inflamed state by inflammatory signals is countered by these signals that reverse it to original state. And the study of this signal, of course, this is just, I'm just proposing this. This is not something that's been studied. We started studying it, and I think there's some examples of that maybe will be, are now supporting this idea. And that type of control, I think, deals with this problem of a trade-off of benefit and cost of inflammation. And but it's just something that I want to put out there, so that if people can find that, if they find it useful in terms of some observations that they make in their different research areas. So I'll stop here. The experimental works that I showed you were done by these very talented people, Andrew, Harding, Sarah. They're all already, well, all of them left the lab already, and Brandon and Fernando helped with various aspects of it, and this is my funding sources, and thank you very much. I'll stop here.
Thank you, Ruslan. It was, as always, I mean, a great way of seeing the world that really makes your research and your input something that is quite unusual. I mean, I think that is very good to
thank you
see your thoughts. I mean, go together with you in your thoughts and in this perspective that is, always using evolution, homeostasis. I think that those aspects are very important. And so we had like people congratulating you and thank you for the great talk and several questions. So I will move to the questions, and if there is time, I will try to make my own question. But Clarissa Maya, I think that we're going to have the questions here. So she asked whether loss of function regulation can be reset of the homeostatic condition, and establishing a new or a chronic homeostatic set point makes it even more difficult to return to the healthy homeostasis.
Yeah, that's I agree with that. So one way that when I mentioned that for each problem, loss of structure, function, or homeostasis, there is this alternative inflammatory counterpart. And one of them that I forgot to mention is exactly that the change of set point is exactly one of the inflammatory states. And that's exactly what happens in sickness behavior. So the body temperature set point changes. So now it's fever. Appetite set point changes. Now it's anorexia, libido, and all these other things. It's the same thing. It's all controlling hypothalamus in different nuclei. But it's exactly right. And the second point is also exactly right, that this change in set point, the ability of the system to change set point is what I think creates vulnerability to chronic disease. For example, insulin receptor has the ability to change set point, right? That's why there is insulin resistance. It evolved not to make us sick with diabetes. It evolved for change resource allocation during pregnancy. There is gestational diabetes, for example, to redirect resources so that mother doesn't consume all glucose and some of it goes to uterus. During infection, insulin resistance allows glucose allocation instead of muscle and fat and liver, it goes to the immune system and so forth. So it's designed to be flexible this way, but that creates vulnerability. And because of the system is able to switch, then it can result in type 2 diabetes and so on. So it's exactly right. It's having this alternative set points is what can create problems in the long term.
So 𝗕𝗃 asked whether eating too much salt, as you also have shown, that drives inflammation. And he want to know how relevant is this aspect in terms of disease in general and the role of macrophages on that.
Yeah, so salt consumption, high salt consumption is one of the features of modern environment, right? It's in nature, for terrestrial animals, salt is usually limited, even for carnivores, but especially for herbivores. But when we overconsume salt, I think there we need to be a little bit more careful about where exactly you would find higher sodium concentration in the body, because there are all kinds of mechanisms to quickly equilibrate it. So that part we don't know, and I want just to say that this needs to be studied carefully, because all we know is that if we manipulate in vitro, high salt induces inflammasome activation and so forth. If we manipulate in vivo by giving high salt water, it induces inflammation in the gut. For sure, it induces Th17 response, and it's also 𝗕𝗃𝗀𝗃, 𝗕𝗏𝗃 showed that in humans. But mechanistically, how it works, I want to be careful that we don't know if this is direct effect of salt or if it's mediated by some maybe intestinal epithelial hormones or something like that. But yeah, high salt. This is a good example of a perturbation that. The other one is pH, and of course, nutrient and oxygen. All of them are different types of perturbations that can result in inflammatory response. Perturbation in both directions, too high or too low, like too high glucose is also very notorious for inducing inflammation.
So Juliana has several points here, but most of them related to meat and dairy product consumption. and she would like to know how these would affect inflammation, and more specifically in the end, whether in viral infection, specifically in COVID, these would have any impact. And that's an interesting point, because like meat consumption, it's something that humans have been doing for a long time as hunters, right?
Yeah.
But then we have this major change in terms of food consumption and lipid consumption and so on. So
yeah,
so it goes a bit in the direction that you have been discussing before with the food and disease tolerance and so on. But
yeah, so with the meat, yeah, we actually have, incidentally, we have a whole project now studying how different types of macronutrients affect the immune system. And the most interesting effects we actually do see with amino acids, with proteins. But these effects are, we don't yet understand them fully, because unlike carbohydrates or lipids, which is kind of a much more homogeneous class of nutrients, proteins, of course, 20 different amino acids, they all behave differently and increased or decreased in different classes depending on whether they're essential, conditionally essential, and so forth, has very different kinds of effect. The type of sort of what we consume in regular diet, I mean, the obvious examples we know of is meat consumption is bad when you have bacterial infection because of iron, and that's kind of an obvious one. But more nuanced aspects of it, I don't think we understand yet enough. Especially, this is the area where using mice may not be
idea,
yeah, because it's such a different. Every animal has different adaptation to different types of diets, and so mice probably not a good model for that. But it's a very important, interesting question. How, because I don't know, but for me, I feel like if I have a flu or severe cold, I always want to eat something sweet. I usually don't like sweet, but only when I'm sick, I want something sweet. So there may be some, we think that there may be some switch in preferences that happens maybe at the level of hypothalamus that controls our food preference depending on the sickness. So when women are pregnant, they, in the first trimester especially, they usually avoid a lot of things that otherwise they would eat, right? Cheese, or there's a lot of things that now for them is not tasty. And I think the same, so there are mechanisms for sure to control preference. And I think they also exist during illness. But we don't know yet what the logic is, that why you don't like this and you
block
like
avoidance. Yeah, you block something that you avoid, and now you start eating and so on, right?
Yeah, and also the same way is like when you're sick, you don't want to have alcohol, for example, right? And why is that? That's probably has to do with detoxification pathways, which are strongly suppressed by inflammation. So when you're sick and you drink alcohol, you feel much more hangover for that reason. So this is one example, but I think it's just more familiar to us. But there are many other that probably fall in the same category, that you don't want some vegetable when you're sick because detoxification pathway is important for it, for example.
So Rodney Bogosian, he asked how to deep the knowledge in the biological story of a critical illness, as you mentioned here. So what would be the next steps in this field, you think?
Well, so first, I think that this is, I don't know if you all agree, but I think it's understudied, very understudied area. If you look at all the medicine, like for almost every class of diseases, there is a lot known about biology, usually, maybe with the exception of psychiatric diseases. But for critical illness, it's all about how to keep the patient alive. It's mostly about keeping, fluids and blood pressure and so on. But obviously, evolution cared a lot about whether you live or die, right? So there must be mechanisms that we just don't study systematically. And I think we need to develop some good framework to study it. Maybe people who come close to this with people who study sepsis. And but even in sepsis field, there's a lot of, I think there is a lack of sort of a common framework to understand this condition. So one, we need to kind of define it,
and also to somehow predict which which patient will have a more severe disease.
Exactly.
In a anticipation way, right?
Yeah.
Anticipating what is going to happen.
Yeah, no, that's exactly right. Yeah, Marcelo. There's in the patients who have in sepsis, as many people know, patients reach the critical point and then they start slowly improving, and then doctor knows, okay, they will be fine. They start slowly getting worse, and then doctors say that probably not going to make it. But we don't know what that critical point is, what determines the outcome. And these are just examples of how important it is to study it. But what I wanted to mention also, that we need to keep in mind that when we study experimental animal, like mice usually, versus what happens to a patient in a ICU, if we want to understand that, we need to use conditions that mimic ICU. For example, everybody who studies sepsis in mice, they don't put mice on fluids IV. But of course, the moment you go to ICU with sepsis, you get fluids IV. And what it does is that the first problem is hemodynamic shock is what would be killing mice. And whatever you do to them, you're looking at that problem. But for patients, that's not the problem. They got fluid IV. They now deal with the next problem. And you cannot compare what you're studying here with this, because different diseases. So this sort of thing, we need to just clarify so that everyone on the same page. Otherwise, there's going to be hundreds of papers showing one thing and 100 papers saying that it's wrong. And that's just silly, right? We need to just have this kind of a framework everyone agrees on, that what are you studying under this condition versus these conditions.
So Bruno Diaz mentions that cetoacidosis, besides being a consequence of dysregulated metabolism, in poorly controlled diabetes can also be less ditch protective effort against glucose toxicity, and such dysregulated mechanism.
Yeah, we looked at ketoacidosis, and similar to glucose, and I'm sure it would be similar to many other metabolites. It's the wrong way to think about them would be whether a given, whether a given level, whether a given metabolite is good or bad. The right way to think if a given range is good or bad, because there's always range. So for glucose, as I mentioned, if you too much glucose causes this neuronal damage, too little glucose will cause other type of damage. So obviously, it's not like is glucose good or bad. This is a silly question many people asked after our paper and Miguel's paper came out and say, oh, you said it's good, and they said it's bad. No, nobody says it's good or bad. This is completely wrong way to think about it. With ketoacidosis, it's the same. In a certain range, ketones are protective. But when the range of ketones is too high, leading to ketoacidosis, then they become bad. They have bad effects again. So if we put on ketogenic diet mice for 2 weeks, after that, they have ketoacidosis and they're very vulnerable to inflammation. So it's always we need to think about the range, appropriate range of these metabolites.
Clarissa has, did you observe any link between GDF-15 and insulin signaling in sepsis?
Between GDF-15 and insulin signaling, we did not look specifically at that. What indirectly we looked, when we block GDF-15, we tried to analyze what happens to why mice die. And when we studied that, we looked at metabolites and insulin and glucose and so on, and they were not affected. So from that, at least based on that, I would say it's not affected. But we did not directly study if GDF-15 has impact on insulin signaling.
Let me see, hold on. I lost a little bit here. There's so Lucia Facioli. She says, we have recently shown a close relationship between IL-1 beta, PGE2, acetylcholine, and cardiac dysfunction in scorpion envenomation. Have you already investigated this relationship in sepsis?
No, we haven't looked at these specific signals in sepsis. In terms of cardiac dysfunction, we only looked at in the context of this disruption of triglyceride supply to the heart in sepsis. And I mean, this also suggests that there are when we probably need also to be more careful and specific about when we say inflammation, like what type of inflammation under what conditions, because as this example illustrates with the scorpion venom, the problem would be, as you show, IL-1 and PGE2, but in maybe in toxin infection or viral infection, it would be different drivers of inflammation, but they're all not all the same, obviously. Like interferon and IL-1 beta are usually antagonistic even. So, I think that's something that we also need to, as a field, agree on, and to have some ways of classifying different types of inflammatory response, because current classifications, I think, are insufficient. They're too old and not adequate.
Yeah, we are still waiting your book, right, on inflammation.
Yeah,
you know that.
Yeah.
So Jones Ribeiro is asking, so he thanks and say, I was wondering about the aspects of GD15 in the case of viral infections in which glucose uptake seems to be beneficial for the host.
Yeah, in viral inflammation or infection, GDF15, so GDF15 was protective to both viral and bacterial inflammation. And GDF15 did not affect glucose. And it also didn't affect fatty acids, only affected triglycerides. And that's why we started looking in the liver and found this sympathetic connection. So the way it's protective from viral infection, I think, has different mechanism, not related to glucose, not different from what I showed in the early part. The early part, that mechanism had to do with very clearly with the damage to brainstem through ER stress. So that we found that if we delete CHOP, which is transcription factor causing apoptosis, it's completely protected mice. So that's a distinct mechanism. And again, I want to say that if we look at, we all appreciate human organism is very complex, and there are many things that can go wrong, and they can go wrong in different order and by different reasons. And so one can die from multiple problems. And when we say something is protective, we generally just say it's protective from sepsis, but more accurate would be to say it's protective from if this problem is
damage.
Yeah, if this problem kills you and you protect this problem, then it's protective. When somebody else studies something else, one step behind or before, that would be different.
And yeah, so he goes on and asks about the evolutionary aspect in the case of anorexia in this particular case, in terms of viral infection.
Yeah, with viral infection, this is something we don't fully understand. There is what we can say is that that glucose, at least in the context of acute viral infection, or I would rather say acute viral inflammation, was protective, and in sepsis, bacterial inflammation was detrimental. But again, the way we should think about it is that what is the limiting factor for survival, right? if, let's say, if the virus is bad because it causes very strong inflammation, and then perhaps promoting tolerance to inflammation is all you need. But if virus is bad because it causes inflammation, but also causes damage to some cells or tissues, then you can die from that, not from glucose and changes or this type of inflammatory damage. And without knowing it, we cannot just say that it's protected from viral infection or detrimental or in a given type of infection. It always depends on what is used. That's why I prefer, as a first step, I always prefer not to use pathogen. I prefer to use something that mimics it, like 𝗕𝗲𝗂𝗶𝗁. People say, oh, this is not real infection. Yeah, of course it's not real infection. I'm trying to understand first the baseline. What happens when the system doesn't have 10 different unknowns and complications. Once we know that, then we can, on top of it, put some specific pathogens and to see how the effects are different. And this is something, again, that is in the field. Some people have strong opinions and also very, they usually have, they think that they're always right. I think we need to agree that, before you study something more complicated, you need to understand more simple scenarios.
Yeah. So Manuela Bastos wants to know how salt interacts with metals in the body.
How salts interact with metals. Well, for salts, by salts we primarily mean sodium chloride, right? So for sodium, I'm not sure there is a big role played by metal interaction, because it usually would be divalent cations that would interact with metals, divalent or more, like calcium and iron. So I don't know if with salt there is an issue. It's more an issue with heavy metals, not with sodium. So I'm not sure that there is anything specific, unless I'm just not unaware of it.
Emiliano Medei says, great talk. And do you think that also could be a signaling crosstalk between GDF15 and some cells from adaptive immune response, like T lymphocytes, in this homeostasis regulation?
Yeah, so this is something that many people now suspect. So the situation is that a few years ago, I forgot what year, but maybe 4 years ago, these 4 papers came up showing that there is a receptor for GDF15 expressed in area postrema. And that's very clear that the receptor is there, and many of these known effects of GDF15 are mediated through area postrema, right? So anorexia, nausea, those are all dependent on that receptor. But also there is a lot of rumors, but I haven't seen a lot of data, but maybe there's one paper only so far. A lot of rumors that there is an additional receptor, and that receptor is expressed in lymphocytes. That data I haven't seen about lymphocytes, but I've heard more than one group that they have such observations. And we also have some data that suggests that there's some local effect of GDF15, not through the brainstem. So I think the answer to your question is that yes, most likely there is this effects of GDF15 on lymphocytes, but so far nobody knows what the receptor is. And I think some of these papers probably will be published sometime soon.
So Fábio Canto say, nice talk. And do you believe that anti-inflammatory and counter-inflammatory signals are always dissociated? Are there evidence that a single component could play both roles?
Yeah, that's a good question that I forgot to make that point. I think that there are some signals that perform both functions. For example, I think TGF-beta and glucocorticoids would be examples that have both anti-inflammatory and counter-inflammatory effect. IL-10, on the other hand, is just anti-inflammatory because the receptor is only on hematopoietic cells. And then the other example that I find very interesting, the one we study more closely, is about adenosine. So adenosine is also made by inflamed tissues, by hypoxic state, when tissues are unhappy. And it has, but it has both effects. It has anti-inflammatory effects through one class of receptors, and it also promotes this kind of tissue adaptation. So something that would fit with counter-inflammatory properties. So yes, I think some signals can do both for sure. And the source of the signals, I think, usually likely to be tissue-resident macrophages and Tregs. Those are probably the two types of cells that produce them.
So someone with the funny nickname Big Boy is asking, in the case of cancer, when tolerance mechanisms sometimes improve tumor survival, do you think that reducing tolerance, for example, by metabolic alterations, could help reduce tumor burden?
Reducing tolerance of tumors. Yeah, if we, yeah, no, for sure. Yeah, I do think so. In general, the question is whether these tolerance mechanisms would be subject to the same kind of mutation and evasion and selection, or if we can find the types of tolerance mechanisms that tumors cannot mutate because they're essential. And if so, whether this would also affect normal cells if we target them. So that's where it becomes tricky. But I think that one example where this tolerance of tumors is particularly bad is one is with cancer stem cells, where they have low proliferation. They rely more on lipid oxidative metabolism. So they're much more resistant to stress, to chemotherapeutic drugs, and probably to immune therapy also. And these types of, and these are, of course, the worst cells because you cannot get rid of them and they can come back years later. And if we learn how to deal with them by actually making them, paradoxically, I think what may need to be done is to promote their proliferation, because if they start proliferating, then they become vulnerable. And then you kill them, and that could be the way to do it. Yeah, so that's how I think one can kind of get them out of tolerant stress-resistant state when they're more dormant, sort of stem cell-like, and get them to proliferate, and then use the therapy against them.
It's similar to HIV in certain cells, right?
Yeah, exactly. Yeah.
So 𝗕𝗏𝗂𝗍𝗂 says, great talk. Parasites also have mechanisms to change host behavior in order to continue their life cycle. Can you also comment on that?
Thinking about that, my favorite but maybe depressing thought is that we know that parasites and even other types of infections can change behavior and so on. But I think that we all already have changed behavior because we all have something, right? So our behavior is not normal, it's already changed. So that's a maybe depressing thought, but we don't know. Maybe normal is better or worse, but we probably all already changed in some ways because...
Or maybe normal is to have parasites, right?
Yeah, exactly. So normal is to have parasites. We don't have them, and now we have depression and unhappy and so on. Or maybe we're more happy. So I think this is a fascinating question, but the question is, what is the baseline? What is the normal, right? And that is hard to tell, because now we're learning how much effect. The reason I started thinking that is because of this possible connection between viral infections and chronic fatigue syndrome. And long COVID is another example of that, where one can say with chronic fatigue syndrome, there are many gradations. It can be mild, it could be very, very severe. Same with long COVID. And when people say they recovered a little bit, but not entirely, if you just extrapolate that to say we all had some bad infections in the past, who is to say we recovered completely or not? So in that sense, I'm saying that maybe we're all affected by every infection we've had before to different degrees. Some of them negligible, but some of them maybe not so negligible.
And maybe some infections including the ability to affect aging cells, right? They might accelerate aging. And
so the biological age, it's a very different rate. You can see two people 80 years old, one can play soccer, the other cannot move without any other disease, but a very different rate of aging also could be. So there are all kinds of interesting questions there that we normally don't consider because we think healthy, infected, but it's probably not like that. It's there is no healthy, like completely healthy. We all, even if not infected, have some maybe consequences. Maybe they're good, maybe they're bad. We don't know. We don't know what the baseline is. So the examples with parasites that are more sort of dramatic examples, those are very super interesting. But maybe they're just showing us the extreme case. But maybe we all are less extreme case with the way we behave. So if we do something bad, we can blame those pathogens.
So 𝗕𝗃𝗀𝗃𝗃, could you comment if omega-3 polyunsaturated fatty acid and/or endocannabinoids have any role, short or long, in the survival effects promoted by GDF-15 in the brain or in periphery?
That I don't know. We have not looked at that at all. Generally, these polyunsaturated amino acids have anti-inflammatory properties, of course, and in that sense, they are presumably helpful. But whether they're connected with GDF-15 production or signaling, that part we don't know. I mean, the most well-known drivers of GDF-15 production are conditions of cellular stress, something that is associated with reactive oxygen generation, mitochondrial dysfunction, activation of NRF2, which is one of the sensors for ROS. And as long as that pathway is affected, and omega-3 fatty acids can affect it negatively, it actually presumably would be interfering with GDF production. But we don't know. We haven't looked at that directly.
Vanessa Pinho. Hi, Vanessa. Could GDF-15 be a pro-resolving molecule? What do you think about that? Are there some studies?
That's a good suggestion that it may have pro-resolving role. What I would say that we don't know the answer because we don't know if there is what the additional receptor is and on which cell types. So as soon as that is worked out, I think there would be many studies addressing this role. But I would say, based on just our experience, I think you're right. I think it has some pro-resolving. So this would be local effects of GDF-15, not the ones that mediated by area postrema. And I think it likely have this kind of a pro-resolving and also tissue protective effects.
So Miriam Werneck is saying, amazing talk. Can't stop thinking about the neuroimmune endocrine axis. Do you think that some of the long-term effect of COVID may come from unresolved subclinical inflammation, some neural side effects?
Yes, that's a very very likely. I would say yes. And this subclinical unresolved inflammation or unresolved switch in the state of some part of the system, some cells or tissues that are switched to inflammatory regime but didn't switch back, or or actually kept actively by unresolved inflammation because there's some residual virus, for example, that could be contributing. Because there are two theories now about long COVID. One is that there is a persistent reservoir somewhere, not detectable by tests, but somewhere, maybe in the brain, in the heart. And the other is that it's autoimmunity, that autoantibodies that have been shown induced in long COVID patients, and that it's an autoimmune disease, similar to EBV and MS connection. Right? So either way, there would be inflammation, right? Either autoimmune or persistent infection. So I think the answer is very likely yes.
So I think that I'm going to be able to make a question here, because I think that we are almost there. So I was intrigued by this idea of a type 1 inflammation or type 1 immunity associated to inflammation that has structural damage. And when we see to this type of inflammation, it's exactly the inflammation that causes more tissue damage. If you compare to allergy, and you propose that, and when you think about, allergy in the skin and so on, once it's gone, it's completely gone. I mean, the tissue becomes pretty much similar to what was before. Maybe, of course, in some more chronic responses, such as like long-term asthma, you have important changes in tissues. But how would you would conciliate that? So it's kind of funny that type 1 is actually the one that causes more tissue damage, and is the one that would be triggered by structural changes in the tissues.
Yeah. No, it is an interesting question. That you're exactly right with type 2 inflammation, and something that I always thought pretty remarkable with allergy, how unique it is as a disease. Because if you remove the allergen, you're just completely fine. There's no problem at all, right? So it's not, I don't even think that it's right to call it a disease, because it's just it's like pain. Pain is not disease. If you hurt, you have pain. And of course, chronic pain would be disease, but it's not caused by whatever pain is designed to deal with. Just like your example with asthma. Yeah. So with allergy, it's interesting that all you need to do is to remove the allergen, and maybe so the question becomes why damage-induced inflammation also is the one that causes collateral damage. I don't have a good answer. My only maybe hand-waving answer would be, if imagine if there is a house and let's say the roof and 2 walls are broken, so there's damage. How do you deal with it? You need to bring bulldozer and to get rid of everything else and then build the new house, right? You cannot just rebuild half of the house if half broken. So under those conditions, first you need to clear it, and then you're ready to rebuild. So for regeneration of tissues, if regeneration is attempted in the context of this half damaged tissue, some cells dead, some whatever, it's probably not an efficient way to do it, just like with rebuilding. And then you need
but it's kind of funny that we always say that M2 macrophages are the one associated with tissue remodeling. You know what I mean? Like, it's kind of contradictory if you think about it.
Well, I think that's exactly so. When you have tissue damage, or if there is a loss of structure, it also results in loss of function and loss of regulation, because without structure you don't have it. So when there is tissue damage, first you need type 1 inflammation to deal with that. But after that, you need to restore the type 1 inflammation to restore structure, but then you need to restore function. Then you need type 2 inflammation, and then you need to restore regulation, and that would be this third one. So they probably all happen in this succession for that reason. But that's just how I think about it from this particular perspective. And yeah, so our knowledge about the repair and the restoration is very, I would say, still very primitive.
So, Ruslan, I again want to thank you, and it's of course a pleasure to see you in good health, and I hope to
Thank you.
to have you around soon, and all the people here, and many people thanking you and saying that was an amazing talk, and I think that
Yeah,
we'll take this home, and we're gonna be thinking about your conference and your
Thank you.
ideas as we have been doing in the past years. So it's great.
Thank you very much. I hope next time I can come finally to Brazil, and I see like a lot of friendly names and faces here. Sorry I could not respond to directly on typing the comments, but thank you all very much, and I really hope to see you all soon in person somewhere in Brazil. Thank you, Marcelo.
Thank you very much, Ruslan. It was great.
Thank you. Thanks, everybody.
Yeah.
Bye.
Bye.
So, thank you, Dr. Ruslan. Was great, the talk. I loved it. Yeah, I think fit well what we want for the closing ceremony. So thank you very much. Well, so this is the end. So now I'm going officially to close officially our meeting. So I ask you to hold on a little bit. I won't take too much. I will say a few words, but some remarks that I think it's important for our meeting and for the perspective of our meeting. So I brought some slides. So I have to say that the meeting was, my opinion, was great. So we got outstanding speakers, great data, good discussion, good audience. So for me, it was a success. So I'm very happy. I think we made it. So I have to acknowledge a lot of people. As I told now, I would like to acknowledge all the speakers for the time, for the opportunity, for to share the data with us. So thank you very much. I hope that next time we'll be in person. I think we can engage more students and we can interact much better than now. So I have to ask to thanks the chairs. I know that is not an easy task. We have to keep an eye on comments, on private chats, time, see the presentation, ask questions. So it's not an easy task, it's multitask. So thank you very much to share this with us and to share our speakers. So it was great. Thank you. I hope to count on you again next time. Also, the students, I already told they were great. I was very positively impressed with the presentations. And actually, our aim, or one of our aims, is to bring students to attract students to the meeting. So I think we were succeeded. So we got a lot of students. So it was great. It was nice. Thank you to participate. The audience, so we got an audience, so it was great also. I'm so happy. I have to confess that I was afraid to have few people in the audience, but it was not like that. So I will show the stats. We got the points. And also I want to thank the Syntagma company. They gave us a lot of support and all the transmission. I think everything was 100%, was fine. So thank you very much. Also, I want to thank again 𝗕𝗲𝗄𝗃. We could make it because 𝗕𝗲𝗄𝗃 gave us a grant for it, for the presentation, for the Congress. And this is the reason that it's a free event. We had support for it. Also, and last but not least, I want to thank the organizing committee. Again and again, we were a team. We are a team. We were together until now. We are a team. We make it together. We share tasks, and there were a lot of tasks, and we could finally prepare and present and offer this nice event. So here is part of them. We were 12, so I won't nominate everybody, but I did this in the beginning, in the opening session, so thank you very much. Here's some before pandemic. We agglomerate a little bit, but before pandemic. So thank you all to be with me here. So some stats of the meeting, we had 1,366 registration. Remember, it's a free event, so we got a lot of registrations. The majority from Brazil, of course, but we got some international registrations. That is nice. We hope to increase this registration for next meeting in 2022. The average attendance on live stream was 300, so was great. So in my opinion, it's a good number. And like we peak in 380, so it was very nice in the opening and in the closing session. So here we can see in this table the median of attendance. So you can see that we were above 200, 250 all the time. So very nice number. We got 7,600 views, so very good. And it's nice to say that almost all countries we got people looking or visiting our website, so this is nice. We got 89 submitted abstracts, and 87 accepted, and we gave 23 honorable mentions. So our stats are very nice. We hope to increase these numbers for next meeting. I hope that next meeting probably will be hybrid. We will be in person and also in a remote version. So I think we can gather more people. I want to remember also that our site is active and will be active until the next meeting. So a workshop on inflammation.org. So visit, leave comments. We are going to feed our site all the time, and we'll begin actually the disclosure of the next meeting with information. So keep an eye on our site. So I would like you only to remember the students that were awarded. So our winners for the oral presentation: Fernanda Neves Cunha, 𝗕𝗲𝗂𝗃𝗵𝗁, Isadora Ramos de Andrade, Luciana Tavares, and also the most visited abstracts: Gilson Dornelles, Isadora Ramos de Andrade, Marina Valente Barroso. Congratulations. So I could not talk, or I have to take this opportunity to say that we are experiencing a very, very hard time in science in Brazil. We are struggling to make science with no funding or shortage of funding. This is very hard for our young researchers, for our students, even for us. So we need to change this. I think we need to say yes to science stronger. So the reason that we are presenting or offering this meeting is resilience, strength, and the will to continue doing science, and good science, and high-level science. So we have to say yes to science, yes to funding, because we have been getting bad news since 2015. Last week we got another bad news about funding from CNPq. So this this has to change. We need to go on. So yes to development, and yes to vaccine, to everybody come back and get our lives not as before, but maybe better. Who knows? So my last topic, I would like to invite you for the 4th workshop on inflammation 2022. I have said that we created this meeting in 2011, was our first event. And then we could only repeat in 2018, and we decided to make it biennial, every other week, every other year. But due to pandemic, we changed it. It's supposed to occur in 2020, but we changed it to 2021 due to COVID-19. But we are going to stick with the plan. So we are going to offer the meeting next year, 2022, as I told before, in person. And we want to this time make this meeting with a theme. We want to praise inflammation. So the idea is to talk about the past, the present, and the future of inflammation. We have a very nice history about inflammation. We have high-level groups in Brazil, and these groups today, that nowadays, came from founders of inflammation in Brazil. And actually, we had almost 2 Nobel Prize in inflammation. So I think this history should be, we should tell this history to our society. And we plan to make a special session next year. And I will invite Dr. Bruno Dias. He will say some words about this. And I will finish here, and Bruno will close the meeting. So thank you very much to be with us, and I hope to see everybody next meeting. So Bruno, the floor is yours.
Thank you, Claudia. It's really a pleasure and an honor to be able to make some concluding remarks and prepare everybody to next year's workshop. So first, I would like to thank Dr. 𝗕𝗂𝗶𝗁 and all my colleagues at the organizing committee of the 3rd workshop on inflammation for putting together a very exciting meeting. This meeting was postponed and forced into a non-familiar remote format, but it managed to keep up with the quality and excitement of its 2 previous editions. For the next edition, we chose to recount the past, present, and future of inflammation research in Brazil. It will be an opportunity to retrace the roots of inflammation research and remember its pharmacologists, precursors, such as 𝗕𝗿𝗂𝗀𝗇 𝗬𝗹𝗂𝗃, Sérgio Ferreira, 𝗕𝗏𝗃𝗃, 𝗏𝗏𝗃, and others, including many international collaborators, such as Sir John Vane, that made impactful and long-lasting contributions in the description of autocoids and the cascade of events in inflammation. These contributions influenced and in many cases contributed directly to the production of many different new generations of researchers in inflammation. Some of those were in display during these events, such as Mauro Teixeira and Patrícia Bozza, but it also includes many others, such as Carlos Alberto Flores and 𝗕𝗏𝗃𝗃, that are sadly no longer with us, and many that are prominent and highly active researchers in inflammation, such as Fernando Cunha, Marco Martins, Patrícia Silva Martins, and Hugo Castro Faria Neto, that continue to carry this tradition and participated in previous editions of the workshop and will participate in the future. In the 4th workshop on inflammation next year, we will be able to hear the full story of inflammation research in Brazil and how it connects with what is currently being produced in our labs and around the world. Furthermore, on how it connects with different areas such as cancer, parasitology, and chronic degenerative diseases. When the world realized that we were facing a generation-defining pandemic early last year, Inflammation research in Brazil and around the world joined the global efforts to tackle the problem and advance the scientific knowledge in a pace never before seen, with tangible results such as vaccines that helped us survive the challenge, albeit after claiming at least 5 million people worldwide. This pandemic highlighted how the recognition of the role of the inflammatory process and eventually its dysregulation in many pathological conditions turned inflammation research as a convergence point for many different research areas that interact and feed each other to advance our knowledge of fundamental biological processes. Once again, all these advances will be on display next year in Rio de Janeiro. In the name of the organizing committee, I invite you all to come and enjoy this rich atmosphere of science, culture, and natural beauty in, as we locals call it, Cidade Maravilhosa, the wonderful city. I hope to see you all in Rio de Janeiro next year. Thank you very much. Thank you for attending. Thank you for participating.
So, thank you.